Thank you. Good morning, everybody. Thank you for getting up early to come listen to me. Before I start, I'd like to thank Tom Ostaiska, I practice that, Morton and Anton and their team, because the organization has been impeccable, which makes the conference so much easier to just simply enjoy, and it's been really, really wonderful. So thank you. It's an honor, a privilege. I'm grateful for the opportunity. So I had to go back and look at my abstract because Tom was, you guys experienced that, like, I need the abstract, and it's been several months. So in trying to honor what I wrote in there, I've been working on the talk throughout the day yesterday, and after hearing every session, I'm like, I need to modify this talk. So I kept adding and not taking out, so we'll see how it goes. But first, let me define a coral holobiont and an Idarian holobiont, because I'll be talking about more than just corals. So a coral is an animal, a basal branching, early branching animal, or it belongs to the branch of the tree, and it lives in symbiosis with a unicellular photosynthetic eukaryote, a dinoflagellate in the family Symbiodinaceae. And they are endosymbionts that live inside the endodermal layer of the coral tissue. But with the advent of genomics and molecular biology, we have been able to uncover the complexity of the holobiont, the Idarian coral holobiont, and this is a recent review of all the other large microbial members that have been identified, and there is still a lot to uncover, but I won't be talking about all of them, so I'm highlighting the ones that I'll touch upon the lecture today, the talk today. So the algal symbionts, and this little diagram represents their life cycle. So they live in hospede, but they're facultative symbionts, so you can find them in the water column, and that is important for their life cycle and also for ecosystem functioning, if you don't know, you will find out today. And also bacteria. We have been doing a lot of research in trying to identify how they're distributed anatomically. I won't have time to go into that, but we have been looking at the coral microbiome for quite a while now. And then there is a whole slew of eukaryotes that also live in them. Some of them are photosynthetic. We don't understand their function as much. There is one in particular I'll touch upon at the end. It's an endolithic green alga that lives in the skeleton, and it'll become relevant towards the end of the talk. So this is how I laid out the outline of the talk to try to honor all the things I wrote in that abstract. So this is a figure that I drew for my very first grant when I was going to answer every question possible about the assembly dynamics of coral algal symbiosis. Of course, I haven't answered any of those questions, just a sliver of them. But I think it really helped me kind of structure the talk today. So what you see is the life history of the host and how symbiosis gets established. And we've heard a lot yesterday about different life stages of an organism, especially early life stages where the symbiont assemblage gets established in the different groups, plants and animals that we discussed yesterday. So that's where I started my career, trying to understand the onset of coral algal symbiosis, and after 10 hurricanes, and realizing that was probably the hardest thing to answer. And my students were like, a revolt. It's like 10 hurricanes, I need data. We kind of moved along this timeline, and then we started to look at breakdown of symbiosis, and that's what you see there is the bleaching phenotype, which is the host and the algae no longer working in harmony and then splitting up, and then recovery of symbiosis. So I will start by focusing on the holobiont complexity in an adult coral, and then I'll touch upon breakdown of symbiosis. I had promised myself for many years now that I would not talk about coral bleaching anymore because we did a lot of research on trying to understand the transcriptomics of this, and it was just really hard to give that talk. But being here and with recent breakthroughs, I feel that I have renewed hope in what we can do to address the problem rather than describe the problem. And that's also an apology to say that I'm going to show a lot of preliminary data, but I figure, you know, to have all these great minds that think about applied microbiology is an opportunity I don't think I'll have in a long time. So that's where I'll be talking about coral bleaching. And then if I have time, I will talk about the developmental aspects of it. And that's our last section, and that's when I'll be talking about the jellyfish, because I wanted to show all these to you, especially. But if not, I want to leave room for discussion, so I may skip that whole section at the end if I run out of time. Okay, so this is a picture on the left of a Caribbean coral reef. And that group of corals, most of them are Orbicella fabiolata, which is the most important reef building in the Caribbean. And this is a very unusual view, because what you see there is pretty much like 98% coral cover. This is a picture from Curaçao, and that is not something you see anymore. But we'll be talking a lot about that coral. And I wanted to show this picture from NASA because I think it's very important to remember that very few, thank you, that very few organisms, and maybe corals are the only ones, that you can say they build a biological, geological structure that we can see from space. But things are really dire, and I wanted to share some scary numbers that some of them are outdated. They only occupy, and I hope they stay with you, because we need advocates everywhere, 0.1% of the ocean floor. They harbor, nonetheless, up to 40% of all marine biodiversity. About 1 billion people derive their livelihoods from coral reef ecosystems, and this is the number that is really worrisome, because this is before this last 4th global bleaching event, and only 9 to 13% have been predicted to be alive by 2050, and I'm sure that number is going to have to be adjusted after the end of this year. So we have one shot at best, and that's why I decided to show some of these very preliminary data that I'm going to show you. So the global distribution is tropical and subtropical, and the main thing that I wanted to highlight that I think is really important is that this is driven by sunlight and temperature. We talk a lot about temperature, but I think we need to understand the role of light in the survival, the ecology, and the evolution of coral reefs to really be able to address the crisis better. And that is because of these photosymbionts. So people thought for the longest time that this was a single species, Symbiodinium microradiaticum, and that's when only ecologists were really working in the system. But with the advent of molecular systematics, people started to uncover a vast diversity that is physiological, evolutionary, and then the genus got upgraded to family by my colleague at Penn State, and now we have multiple genera with different physiologies that are tied to light and temperature. So Orbicella is, as I said, fabiolata is the major reef builder, but this is a species complex of extant corals that make up the reefs in a great proportion of the reefs in the Caribbean. And what we see when you go in a reef now, you see the elders. The demography is completely destroyed. You don't see recruits. They're not reproducing or settling. And if you lose one of these colonies, you lose very important individuals in the ecosystem. So understanding the physiology is really important for their conservation. So Annularis is a shallow species, and it needs a lot of sunlight. And Fabiolata is more of a generalist that you find from shallow to deep. and they can work with different light regimes. And then Orbicella francii, it's really deep, and you see the colony morphology adjusting to these light light needs. And as you see, if you look up close, it's very hard to discern which is which. You need experts, taxonomic experts, or a lot of practice to really kind of be able to tell them apart at the, can I point? I haven't used the pointer. If you look at this level, you only francii you can tell apart. So it's tricky. And they also form assemblages with different symbiotinaceae that have different physiological demands. And this is not completely representative of what we know now, but it's just to illustrate that Fabiolata establishes more symbiosis with different organisms. And this is very unusual for corals. They tend to prefer a single algal species. There is 3 species that are extinct today. Annularis and francii diverged 0.5 million years ago, and Fabiolata branched off earlier. And if you look at the Pleistocene, I'm going to acknowledge the younger members of my lab and junior collaborators on every slide, and then everybody else will be acknowledged at the end. So Carlos was a postdoc in my lab, and he wanted to understand, and he's still studying this, the evolution of the family. But this graph represents the species richness in corals in general in the Caribbean, and you see a dip around this time, and that's where there were a lot of extinctions because of sea level, changes in sea level. And this is now the fossil record for Orbicella, and each color represents different lineages. So you see there was indeed a lot of extinction in the Pleistocene of many lineages you no longer find, and there were only 4 groups left. And then Orbicella nansa is now extinct too. They went extinct approximately 100,000 years ago, and they have a very different morphology. They're similar to Fabiolata, but Fabiolata has the skeleton here and only the live tissues at the top, whereas the Nansii had live tissue all over. So they had similar ecological niches, but not exactly the same. If you look at the fossil record, what you see is that it used to call the organ pipe Ontastria because there's been some taxonomic revision. So now the genus is Orbicella. And in the Pleistocene, the organ pipe was the most important one. And then Annularis was also very important in the shallow. This is the depth distribution here. And we were able to sequence a few genomes and use coalescent theory, and I won't have the time to go into all the details of that, and it's Carlos's expertise, not mine. But basically what we were able to see is that there was a population expansion at the same time as in the 2 species that like more light in the shallow. At the same time, the organ pipe went extinct. And the paleontologists, this was a beautiful paper because it included all the people that have been working on the system from the paleontology and taxonomy groups. And they were delighted to see that the genomic data kind of corroborated what they found. So they colonized the shallow niches. But the Orbicella francii that are found in the deep were not they didn't change. They didn't have this population expansion. And these colors, I will use them throughout, brown, red, and blue. So this, we have known from the fossil record, this distribution, but I think it's primarily driven by light, and it's something that I want to hammer down because a lot of people think about temperature today and not light, but light is critical. And light comes in very different types of flavors because this is my cheat sheet. I work with coral physiologists, so I'm not one. The attenuation coefficient depends on light absorption, but also scattering, so how clean the water column is. So this is our 3 different reefs in the Caribbean with different light attenuation coefficients. So that can push lower or deeper the distribution of the species. And now this is a collaborator, a young scientist from Colombia, that has been looking at the distribution in different reefs around the Caribbean. And the distribution is the same, but the most important thing for you to notice is how the depth distribution changes, and it is correlated to light. And we have performed a series of reciprocal transplant experiments all over the Caribbean using these as a foundation of how we do the experiment. I will give you one example. So this is a reef in Panama at the Smithsonian field station there, and we chose to work there because Dawn Levitan and Nancy Knowlton have been working there for decades now, and they have mapped every single colony in this transect, and they know who they are. So there is very few Fabiolata there, and in fact, that colony is a genet and is gone extinct from the reef. But we did a reciprocal transplant between Franksai and Fabiolata annularis, and these are the young people that did the work. The coral evolutionary biologist, Joe, is the microbiologist. I'm going to start to get into the microbes, non-dinoflagellate microbes. Joe is the microbiologist, and Tomas is the physiologist. So this is an example of what we do. So we grabbed Orbicella franksai that are found deep, and we do a reciprocal transplant of Orbicella at the same depth, and then they go to shallow, and then you do the same for the other one that is shallow to shallow, and then shallow to deep. And what we observe is that there is a lot more mortality when you deprive the holobiont from light. I'm going to jump into the microbial data very soon. So when you look at photosynthetic physiology, you see that the franksai actually fare better when they move to the shallow. Even though there is more pressure, UV light, and things like that that produce ROS, they do a lot better when they get more light. But when you do that with annularis, they really don't perform well, and it's an unsustainable symbiosis that leads to higher mortality. And this is one slide of how we looked at the microbiome, and things started to emerge that, really popped out. And the annularis, when they go to deep, they really experience a dramatic shift in microbiome composition. And this is an example of some of the groups that changed, and most of them in this particular experiment are upregulated. And then we started to think about how is light also affecting these microbes that are associated to this particular holobiont. So in the series of many reciprocal transplant experiments that now we've done, this is a summary of over a decade of work, and this is the work of Colin Howe, a PhD student. He's looking at, what is happening in terms of the microbial assemblage when you change light regimes, and you also have to keep in mind the evolutionary context, because these are different, closely related but different species. So you have some things that are changing, going up in abundance, and some of them are going down. So here, for instance, you see that from shallow to deep, Endozoicomonaceae that are thought to be symbionts, although the jury's out on that, and Synechococaceae are going up in abundance from shallow to deep, but if you look from deep to shallow, it's very different. And the Rhodobacteriaceae is the same, depending on the direction, but some of them also change from shallow to deep. So the host context is really important, not only the light regime, and some of these are photosynthetic microbes. So how is light affecting the symbionts of the symbionts is something that now we want to try to get at. So this is something that I've been so jet-lagged. I haven't slept for many nights. I was like, okay, I need to update this because it's super relevant. So this is 2 days old, this chart, and I want to call your attention to 2023. So these are sea surface temperatures, and you've heard about this in the news, I'm sure. Way above, it was an El Niño year, so it was particularly hot. And this is 2024. And since last year, the 4th global bleaching event has been happening, and I'll tell you more about that. This is an example of Orbicella from the US Virgin Islands. So it started in a little over a year ago. This is the regular phenotype, and then they go really, really pale, and then they start to recover. And this is in spring. So they can recover, and they do recover, but there is a lot of mortality that has been happening. And there is many threats to life history of corals. Temperature is the one that we talk about the most. So this is also I just downloaded these from NOAA. And I don't think I need to explain it in a lot of detail, but the level of alert, it's linked to degree heating weeks, so how many weeks the temperature, sea surface temperature, is above what corals are expected to tolerate. So you see as Terry Hughes says, he's a scientist in Australia that is a strong advocate against carbon emissions and very vocal on Twitter. It's fun to follow him, and he's really a strong advocate. He's like, there's nowhere to go, there's nowhere to hide if the water gets warmer. It's basically the bottom line here. And that's what brought me to, now this is the scary part of the talk for me, because I know many of you are experts in ancient DNA technology, and I am not. But my naive knowledge has improved from getting beaten down by real experts when I went up to them with my first idea. If you walk around the Caribbean, every church and wall and everything is built with Orbicella, in particular Orbicella faviolata. The Spaniards were, building these fortresses with these corals. And I kept thinking as I went to different sites, I wonder if we can sequence this. I wonder if we can sequence this. And then, it led to this. So we've been taking cores out, but these are examples of cores from different corals. And in fact, I think these are all from the Indo-Pacific. But coral cores are like tree rings. You can date annual events, and then you can pair them with geochemical proxies, and the geochemists are particularly excited about this, because then you can look for signatures of context, environmental context. So in 1998 was the first global leaching event, and you can pick it up in these coral cores. Storms you can also detect, and freshwater anomalies, more water runoff coming from the shore. And in the Indo-Pacific in particular, you can go back 400 years. So we have this incredible record of what has happened in the Anthropocene. But it's not just temperature. There is all these other life cycle threats, and this is from a report that was required from NOAA to add. There used to be 2 species of corals that were listed as threatened. Now, after this, there were, I think it's about 70 or 80 more, all of them in the Caribbean. And these are all the different things that can also affect the survival of corals. In particular, ocean acidification, seawater chemistry affects coral calcification. Sea level rise also affects the coral-algal symbiosis. But then everything else all affects the holobiont as a whole, especially the microbial component. More sedimentation, turbidity, nutrients, eutrophication. These are usually oligotrophic, nutrient-poor environments, and a lot of pollution from sewage and that kind of stuff. So that took me to, I always wanted to do a project at home, and doing genomics, it's tricky because you want to be aware of the social context. So I went to give a talk maybe 10, 15 years ago now at a zoology conference, and the Colombian scientist said, you need to see this reef. And it's called Varadero, and it's near Cartagena. Cartagena de Indias. I was just in Spain, and I saw on the boards there is one in Spain. It's the only place in the world where before you board for a flight to Cartagena, you need to check Cartagena or Cartagena de Indias. So in Cartagena de Indias, there is the reef, Varadero Reef is right here. It's the last man standing, because when the Spaniards came in the 1600s, they diverted the Magdalena River into this channel. Cartagena was a really, really important port before they did the crossing to the Antilles and then to Europe. So getting the goods to Cartagena was really important. So this freshwater input was created several centuries ago. And this is a map of that. This is the city. Also, the sea level has changed, and there is a lot of good historical records and a lot of corals in all these walls, and that's where I would like to go next. This was really important. This is called Boca Chica, the only place that gave access to the city, and the city was attacked by pirates a lot through Boca Chica, and here is Canal del Dique, okay? So in the 20th century, the Colombian government turned the Canal del Dique into an irrigation channel that kept being widened and widened, and in the 80s, the input of fresh water was huge, and now we can detect that in the cores. So we took this one core out. We've taken many more, but I don't have the data to show you, so I'm just going to talk about the one core that we then use ancient DNA approaches to look at the holobiont. And I was fixated on the idea of, oh, we're going to see these endolithic algae because we know that they play a role in coral bleaching. So because the importance of Cartagena, I won't give you a lot of details of this, but this is the different major historical events that we want to test with geochemical proxies and pair it with the ancient DNA technology. And when I was here last year, serendipity, I was talking to Rafael, who's a postdoc with Anton here. We were at the end of the conference, on the bus ride to say goodbye. We sat together, and then we started talking about this, and it's led to a really wonderful collaboration and potential career shift eventually for Rafael. He's helped us with he's the actual ancient DNA expert, so in the group. But this is the preliminary data from this one core. So here you have all the reads, and I'm going to break it down, just focus on these two. So this is the eukaryotic fraction. Most of it is coral, which surprised me, but I'm excited about. And then Symbiodinaceae and Ostrobium. And Ostrobium has been linked to blooming when corals bleaches. They're poor competitors, so they push down in the holobiont, but then they bloom when the coral is white, and that helps in photoprotection. And the coral doesn't die if they have Ostrobium in good quantities, because they're there taking up all this light that otherwise would be damaging, creating ROS and all the reactive oxygen species and so on. And this little blip of Ostrobium data is really exciting, because in 2005 and 2010 were the two last really bad bleaching events in the Caribbean until this year, and we are picking up signatures of that in the record. So this is Ostrobium. This is a cross-section. This is not Orbicella, it's a different species. You found them all over the world, and that's why this is really cool. And then when the coral whitens, then they increase in abundance. And then you can also uncover the diversity of Symbiodinaceae over time, and we know, as I explained, they have different physiological adaptations. So we can look at the temporal light dynamics in the reef and link it. I know it's a correlative analysis, and sorry, Thomas, but that's what we can do so far with what we have in hand. But I think it's really exciting because we can get into the historical ecology of the Anthropocene with crucial organisms. And unfortunately, this was a pilot, and I've been working with the ancient DNA people at Penn State, and they're super thrilled, but when we went in, they say 60% of the projects that people start with them fail. So we were cautious how much we invested. But so the read representation is not great for every time point, but now we know it works. And to give you a little bit about the prokaryotes, we were able to uncover 36 mags from this time series, and the time points are clustering together, and we see signatures of polluted environments from the microbiome. So it's early days, and COVID threw us back. The ancient DNA lab was had issues for 2 years. It's been 4 years, but now we finally are getting into sequence. We have spatial temporal gradients with cores in the Caribbean, and now we're hoping to expand to the cores that the Australian Institute of Marine Science has that date back 5 centuries, basically, and be able to look at these and compare recent history with what happened in the last 50 years, which is critical because that's when thermal stress has happened. So this is IPCC scenarios, and business as usual versus business as usual, and then everything else. So it's really scary what is going to happen, and most of the CO2 that we emit actually goes into the ocean. So these are also coral bleaching predictions under those models, and I won't delve into that. What you need to see is that we have one shot at best to address this. And this is all prior, this is 2023. And in the last year, in fact, there is a number that I recently came across from the Great Barrier Reef. Corals that hadn't bleached this year, 97% of coral cover was bleached in some of these Australian reefs. So mortality is pretty high. Okay, so I don't have a whole lot of time, so I want to just give one example of why I decided to talk about coral bleaching and why I wanted to share this with you. This is a summary of an incredible dissertation of a student that I helped a little bit, and this is an example of an answer to the question that Anton posed the first day: how can we make this more of a level playing field? This student from Colombia, who's outstanding, did incredible experiments, but he didn't have access to genomic technology. And he asked me to co-advise him, and he was able to spend 5 months in the lab and really turn this dissertation into something that can potentially be transformative for restoration. So I think microbes is where it's at. People are talking about crazy things like doing CRISPR on corals and experimental evolution of the photosymbionts, and I think now we have to try it all, actually. But you do CRISPR on one coral, and there's 800 species of corals, and they're not going to all react the same way. And I think working with the host we don't have time. We don't. We really don't. So there is things that we know stress corals: UV, temperature, and what he did is he screened all these different things. He did a very, very, very wide screen. People have been trying to do microbial manipulation on corals as probiotics to address bleaching, but the extent of the experiments that he did is like mind-blowing. This is his slide that it's very elaborate. But he grabbed a coral colony, a coral species that is predominant in urban reefs, and isolated things and made probiotics that then he tested as a prophylactic and therapeutic, and he got really good results. And this is built upon the work of many others. But if we can use these approaches informed by what the ancient DNA reconstructions tell us, I think we can address the issue, but we can also use models to informed models. And I'll say that in this summary because I think I'm running out of time. So I think I'm preaching to the choir, but nonetheless, I think that the ancient DNA holobiont perspective is really important for corals because we can now really understand the Anthropocene and predict forward better climate change scenarios, and then use that to inform these crazy things that we're trying today. So I'm going to skip through the last bit. I'll show you, Thomas, later, all these jellyfish stuff. But we've come up with a model system that allows us to do all these manipulations better in the lab, and that's the point, because corals are really difficult to work with in the field, permitting issues, they only spawn once a year, so looking at the early onset is really difficult. So I'll skip through all of this because I've told you most of this throughout, because I wanted to share this with you. Since the beginning of the conference, we've been talking about these microbial extinction that nobody else is talking about but us in this room. And the COP Biodiversity Conference is in Colombia this year. And this was a last-minute situation that developed because Turkey was hosting it, and then they had the earthquake. So they're in the planning stages. There's not a whole lot of time, but I think proposing a session from this group would be really interesting. So we can talk about that. And I just, it's Pride Month, and we've been talking about embracing diversity, so I use that for my acknowledgement slide, and I'll be happy to take questions, if there's time, which there isn't. Sorry.
Well, thank you very much for your work. Given our buffer, if you promise to be swift for the photograph, we will have time for one question while Kevin gets set up. It's too early.
Yeah, we need coffee.
I have one, then a quick one. So if you just think a little bit more beyond the corals, isn't it also really a neat model to, it sounds like you're redefining the phenomenon of phenotypic plasticity beyond just being contingent on the host genotype, right? So despite this ecological crisis, there's a whole lot more you can do with this system.
Absolutely, especially with the jellyfish system. We were looking at the onset of symbiosis with different symbionts because they're very promiscuous in early stages. They take up anything that, the ocean is so, what's the word? Stochastic. So you grab whatever you can and then you do this winnowing process. So we wanted to do that in early onset, and we chose the jellyfish to look at that. And then we saw the incredible developmental diversity in phenotype, strobilation phenotype, which is the metamorphosis from baby to adult, juvenile. So I think that symbiosis is playing a very important role in host phenotype in many ways besides mechanistic ways of establishing the symbiosis. The photosymbiont is actually playing a critical role in life cycle on how the corals, they establish symbiosis and then they start calcifying. So they don't make a skeleton or a very good one unless they have photosymbionts. And the jellyfish don't strobilate at all. So they do not complete the life cycle unless they establish symbiosis. So yeah.
Okay, thank you, Monica.
Thank you.